I guess you can hear me enough. Is that fine?
Yes.
Okay, so good morning to you. For me, it's not morning anymore, but good morning to you in over there. It's very sad not to be present in person, but just didn't work out this year, and I hope it will be next time. But still, I want to, yeah, I hope you will have a nice meeting and enjoy yourself over there or on the other side of the world. And so I'm going to start. I wanted to start with presenting what we did and with the Biological Resources Working Group over this last, what is it now, 2 or 3 years. And I will just talk you through what we did. So okay, so I will talk about our members, the purpose and our goal of the group, and then we'll do a progress report and I will talk about our achievements, and then I will let you know our plans, and then we will just briefly also touch the leadership. So our active members are listed here. So it's a crew of people that showed up mostly. So in our bi-weekly meetings, but the list of other members is much longer, but we decided here to show only the very active members. And the group is co-leaded between me and Johannes Struck, who's also not present this year, unfortunately. And we had a third person, Cinque Soto, but he had to retire because he's now working for the FDA, and so he couldn't do both things at the same time. And yeah, so that's for this. And we had also several members joining during this time, which was very good. So what is the purpose of our working group? So what we want to do is to provide the community with biological calibrators and reagents to evaluate their experiments when they do AIR sequencing. So we have actually 3 blocks where we work. It's the coordination, the reach out, and the development. So on the left, you can see, so the coordination is to help other groups and other entities to develop their reference and standards and samples that we can then use as controls. And then also our working groups, subgroups of our working groups also work on this. We also develop protocols and reference samples, as we will see. I will show you later. And then the third thing is then after this, once we have the standards and we have developed samples and protocols, we want to reach out to the community and redistribute our knowledge there with the idea to encourage people and to make it easier for them to adopt this kind of standards and rigorous protocols. So our goal is, as we said, to recommend a set of biological standards that can be used for normalization of experiments. So this is the goal we want to achieve. So first, as I said, we will generate and share inline and external standards for repertoire analysis. We will develop those with other groups, as I said, and then we will generate data using those standards we have developed and different library prep methods that we then can compare. And in the end, we will share the data, the standards, and the metadata and analysis pipelines, and we can do comparisons and evaluations. So there's actually three big groups, three big blocks of things we want to do. So the generation of data, and the sharing, so that we can, in the end, really provide the community with recommendations and materials to standardize their experiments. So the progress report and achievement. So regarding the first block of things we want to do, so the generation of standards and for repertoire analysis. So what we did recently, so we, Nina and Andrew Farmer especially wrote a small grant application that they submitted to TABS to start a comparison on BCR sequencing methods with different standard templates. This included spleen, DNA, and RNA, and cell line mixtures and different things. So the idea is there to get a little bit of starting money to produce the standards, sequence them in the lab, and then compare sequencing to methods, RNA and DNA, and then have this as a basis then to do larger comparisons. So this is something which is already ongoing. So they have started already to also produce those standards. So this is really something that has finally started. The same thing is planned to be done also on, for TCR. We also hope to have an effort that is similar, and we will base this on publications from Encarnita's group. It was 2021 in Nature Biotechnology, where she compared different methodologies of sequencing and library preparation. We will base ourselves on this and also try to start developing standards that we will then apply with the best methods that were published there. So these are the things that are ongoing. Finally, after a long time of more theoretical approach, now we started really to generate the standards. So regarding the second block, so the data analysis of data produced with the standards, we have nothing yet to show. But regarding the third block, so the data sharing, and there we have some things to report. So we have had a publication in eLife in 2021. So this was basically our starting point, where we said, okay, we need to do a survey and see what the needs are out in the community. And we need to really pinpoint what is required and what we want to do. And there we discuss really the needs and the challenge for air standards and standardization. Second, also a big block of work last year during the lockdown, there was this chapters we wrote for the methods in molecular biology. So this has been accepted, and this has been basically assembled and headed by Anton Langerak from Euroclonality, and our working group has provided 3 wet bench chapters and wet bench methods, and also a benchwork overview. And there we have very detailed protocols and guidance and suggestions, and really we review the current state of the art or for AIRR-seq. So this is something very important for us, and you will see probably that other working groups also contributed to this big edition. Then what's also something what's been happening recently, the group has been, we had an addition of Wenming Xiao from the FDA, and he approached us and started to join meetings and also joined the working group. And he is starting a very similar enterprise, which is calling AIRR-seq, preparing also standards and reviewing medicines and sequencing technologies in a very, very big, it's a very, very big enterprise with many, many groups involved. And so we have also been asked to participate, and this is also something we do. We will be there, partially actively participating, or also just being part as if they need any help from us. And then also the members of the working group participated in some meetings from Euroclonality, and so there is also contact and exchange there for the methods they have and the standards they have. So the plans for the coming term. So again, our 3 blocks. So regarding standard generation, it's continuing to develop the BCR reagents and the TCR reagents to really get this going. And as I said, this is going to be different things: RNA, spleen RNA or DNA, mixes, cell lines, and also artificial fragments. And we will also actively participate in this FDA effort. And then we will, once we have these things, we'll generate data and publish a paper about sequencing depth and sample optimization for UMI. So this is a project from Nina's side, and still also helping in FDA effort to generate data. And yes, regarding sharing data, we'll still try to be involved in Euroclonality meetings, in the FDA effort as well. And then we really hope that at the end of, or closing the end of the term, we'll be able to set up some recommendation for how to use the standards that we will have produced, and the different methods that we will have reviewed. So this is our long-term goal, of course. And that's it. And now we come to the leadership. So this is what we propose as co-leaders for 22 to 2023. So far, we had me and Johannes Truck, and we both are willing to continue to do that, to have some continuation. And then new and it's very nice, Nina proposed to help out, so that we have again 3 people, somebody from the so 2 persons from the BCR side, me being more TCR side, and we as the European part, and Nina will be the part in the US, representing the US. So I would be very happy if this could happen this way. And that's it from my side, and I'm open for questions if there are. And again, thank you very much.